Nowadays, there is a lot of diseases that cannot be cured murders, no fear at all, but because of gene therapy, there's a way somehow cure the diseases. So in gene therapy, it is an experimental technique that uses genes to treat or prevent disease that in the future. This technique may allow doctors to treat a disorder by inserting a gene into a patient's cells instead of using drugs or surgery, because some diseases are caused by errors or mutation in specific genes. So it utilizes a functional copy of a gene where it has to be potential in treating the disease, thus providing restoration and therapeutic benefits. The first gene therapy approved by the FDA was Kymriah for certain pediatric and young adult patients with acute lymphoblastic leukemia. However, long before Kymriah, the first approved gene therapy clinical research in the U.S. took place on 14 September 1990 at the National Institutes of Health under the direction of William French Anderson. 4-year-old Ashanti De Silva received treatment for a genetic defect that left with her ADA-SCID, a severe immune system deficiency. Let's now move on to the two types of gene therapy. Virtually, all cells in the human body contain genes, making them potential targets for the gene therapy. However, these cells can be divided into major categories: somatic cells, most cells of the body, and cells of the germline eggs or sperm. In theory, it's possible to transform either somatic cell or germ cell. So there are two different types of gene therapy depending on which types of cells are treated. One is germline gene therapy. It involves the modification of the genes inside germ cells, sperm, or ova. 2. Unlike germline therapy, somatic gene therapy only involves the insertion of therapeutic DNA into the body cells and not the germ cells or gametes. This means any effects of the therapy are confined to the individual being treated and are not inherited by future offspring. In summary, the distinction is the results of any somatic gene therapy are restricted to the actual patients and are not passed on to his or her children. All gene therapy to date on humans has been directed at somatic cells, whereas germline engineering in humans remain controversial and prohibited in, for instance, the European Union. Let's now move on to the two types of somatic gene therapy. In vivo, genetic material transferred directly into cells within a patient. Term simply means any kind of experimentation or test done is outside the original living system from where the cells have been taken without much alteration of the natural conditions, which are already present and not exclusively made for the sample. In vivo gene therapy means that therapy is administered directly into the patient. The target cells remain in the body of the patient. The gene is being transferred to a patient's cells while still in the patient. However, a gene cannot be introduced directly into a patient's cells. Instead, a vector, which refers to a virus, is genetically engineered to transfer the gene. Question: will the virus make humans sick? The answer is no. Once the virus delivers the gene into the cell, the virus slips away. Certain viruses are often used as vectors because they can deliver the new gene by injecting the cell. The viruses are modified so they can cause disease when used in people. Next one is ex vivo. Cells are removed from the patient, genetically modified, and transplanted back into the patient via IV directly into a specific tissue. Simply means which takes place outside an organism. And note not in original living system. So as you can see in the figure, in ex vivo gene therapy, the cells are being taken out from the patient, and these cells will be modified in the laboratory when the cells become restored or functional. The cells will be now returned into the patient's body. Let's now move on to the recent developments of gene therapy. Researchers are testing several of approaches to gene therapy, including one replacing mutated gene that causes disease with healthy copy of gene. 2. Inactivating or knocking out a mutated gene that is functioning improperly. And third, introducing a gene or a new gene into the body to help fight a disease, although the gene therapy is a promising treatment option for a number of diseases, including inherited disorders or some type of cancer and certain viral infections. The technique remains risky and still under the study to make sure that it will be safe and effective. Gene therapy is currently being tested only for diseases that have no other cures. So to conclude, theoretically, gene therapy is a permanent solution for genetic diseases, but it has several complexities. At its current stage, it is not accessible to most people due to its huge costs. A breakthrough may come anytime, and a day may come when almost every disease will have a gene therapy. Gene therapy have the potential to revolutionize the practice of medicine.